Hello everybody, and welcome to today's SEDS Online webinar. As always, we'd like to thank our sponsors at IAS, which allow us to offer these resources free of charge. This includes our research lectures, learning tools, and even field trips. So make sure you take a look at our website to see all of our new information. Today's lecture is Dr. Leslie Millem, who is a carbonate sedimentologist and professor at Western Illinois University. Leslie received her master's from Western Washington University and PhD at Southern Methodist University, and her research interests include carbonate sedimentology and diagenesis, dolomitization, sedimentary petrography, and low-temperature isotope geochemistry, as well as geomicrobiology of cave formations. So thank you very much for being here today, and I will hand it over to you.
Good morning. Well, good morning from Illinois anyway. I realize not everybody else, it's morning. I'd like to talk to you about some cave pearls I've been studying. I apologize for my voice, and I have a throat lozenge, so I apologize for any problems with that, but colds don't, follow schedules. I want to look at some cave pearls that I've been studying that are actually mine pearls and look at some of the implications that come out of these samples. So this is part of an ongoing research project I've been working on with a diverse group on the geomicrobiology of cave pools, minerals that grow in pools. How do you tell how much of that rock used to be alive? And it is a collaboration between the geology side, which is myself and my co-author, Mike Spilde at the University of New Mexico, and the biology side, who's Dr. Penny Boston at NASA and Dr. Diana Northup at University of New Mexico. And if you're going to work in caves, you seriously need a good cave photographer. And Dr. Kenneth Ingham is professionally a computer geek, but he's married to Diana. And he's a very good photographer. And that'll make a huge difference. You'll see in the presentation when the pictures are mine how bad they are. But mostly what I want to talk about today is a system that doesn't have much biology, but that's part of understanding the spectrum. So some questions that have come out of our study. Many speleothems, particularly stalagmites, are very well studied with annual layers. So here's a couple of the cave pearls from the Quincy mine. And the question is, are these annual layers? And how might one tell? And then recrystallization is a critical issue in speleothems, right? You want a primary signal for your paleoclimate data, but also in lime muds going to micrite. And here I'm restricting my definition of lime muds to the fines, right? The clay-sized particles that end up being micrite in a limestone. These pearls have some questions, have answers for some of these questions. So how much of these pearls is primary? By the end of today's presentation, I'd like to convince you that very little is primary, despite these pearls being only a few years old. So what's a cave pearl? It is on the spectrum of coated grains. They range in size from less than ½ centimeter. I've got some that are as small as 2 or 3 millimeters, smaller than that, and they don't have the coatings, up to as much as 10 centimeters. They were first described in Carlsbad Caverns in a location in the cave known as the Rookery because of the number of little nest-like things full of cave pearls. They are most commonly calcite, but not exclusively. This one from the Rookery actually has a bit of aragonite in it. They usually form around a nucleus, but you don't always have one, and you don't always catch it depending on how you cut your pearl. If you don't cut it right down the center, a small nucleus could be missed. So thinking about how and the conditions for why cave pearls might form, and I'm just using cave pearl as a general term, and mine pearl is the same thing as far as I'm concerned. They need some basic parameters, which are true for pretty much all speleothems. The incoming water has to be saturated with respect to calcium carbonate, and that's pretty standard. If you've got any limestone above your hole in the ground, whether it's a mine or a cave, it'll probably be saturated by the time it enters the cave. For pearls specifically, you need dripping water or slowly flowing water. So this is the spectrum of cave pearls, if you will, from the Rookery on the left, which is a drip zone. So that would be this one, to another location in Carlsbad that we discovered. These are pearls. They are nice coated grains when you cut them open, but there's no drips here. This is actually a very shallow, must have been only 1 centimeter or 2 deep flowing stream. And then my current project that I haven't got done yet, Arthur Singleton Cave in Kentucky, these pearls here are over 10 centimeters. And this is a pool. It does, however, flood every time it rains. So slowly flowing water that can go stagnant. The third requirement, which is what really restricts how many cave pearls we find, is it requires a flat floor. If you don't have a flat floor, you're more likely to get a stalagmite or something like that. So the process of making them is pretty much the same as any other speleothem. The primary drive is CO2 degassing as the groundwater enters into the enclosed mine air or cave air. In some cave pearls, you also have a role for evaporation. You can see from the Arthur Singleton picture that the water line actually goes up to the limit of that dam, and the pearls go completely underwater, and then it evaporates. So you get a little bit of chemistry showing evaporation. And then more or less microbial action. And it seems to be from our studies that the more stagnant the water, the less the water is moving, the more microbial action. And this is consistent also with some of Brian Jones's work. So the rookery has very active drips and has almost no biology in them. A slow flowing stream has a little bit. Arthur Singleton Cave, if I wasn't careful, would attack me. Just total biology. What I want to think about today, though, is a system in a mine that is pretty much pure CO2 degassing. And that helps us understand some of the controls if we can eliminate some of our variables. So here is where the J. Huber mine is. It's in the central US on the edge of the state of Illinois. It is in the Mississippian Burlington limestone out of the type section Mississippian. It is a carbonate ramp that is absolutely chock full of crinoids. It's almost pure crinoidal. They are mining it actually here for the pure calcium carbonate. Other parts of this unit have some dolomite, but this is really the calcite section. Nice and convenient that the mine is only an hour from where I live at WIU, Western Illinois University. Makes it easy to get over to the mine on a regular basis. The key for sort of the speed with which things are happening in this mine is that it is ventilated. Unlike mines, some mines use electric machinery. This one uses diesel front-end loaders and trucks. It's very nice from a viewpoint of my research in that I get to drive my car in and I can park right next to my field location, no hiking required. But this ventilation system, which means that big front-end loader there can drive by and you smell nothing, also increases the rate of CO2 degassing. Because as the water enters the mine and degasses, that CO2 is immediately whipped out of the mine. The other benefit of working in a mine versus a national park like Carlsbad Caverns is nobody cares how many samples I take. Carlsbad limited me to 3 pearls per location. I have literally hundreds from this location. The climate in this area is a pretty continental climate, expect in the middle of the North American continent. Has fairly hot summers, high temperatures around 30 degrees C, fairly cold winters with lows somewhere around -10 C, gets below freezing most winters for a fair amount of time. When we look at the mine temperatures, the upper diagram shows the mine air range as that gray bar. And I actually put a hobo data logger in the mine. I did this cross so I could hang additional things from it. It apparently got a great deal of discussion from the mine drivers as to exactly what was buried there. Notice that the temperature of the mine actually varies, not as much as the seasonal temperature outside. Caves usually hold a pretty flat temperature that's annual, but here the ventilation, right? They suck in cold air in the winter and warm air in the summer. So it's sort of a muted average rather than a flat average. The gap is when I had so cemented in my data logger that I had to take it back to the lab and clean it of calcite. This is a pretty rainy climate. It has rained pretty much all year round, a bit more rain in the summer. Interestingly and sort of useful for the study, site A, and I'll show you what the sites are in just a second, it dried up almost completely. And so we had a drought from 2005 to 2007. 2005 is the first time I entered the mine. And then you can see we have more normal rainfall following that. This sort of helps us see one drought cycle at least and see whether that leaves a signal in the rocks. You'll note I did sample A for a while and then I shifted to sample B. I'll show you why that is. Here is the two locations in one picture. The one on the right is sample A. We sampled that one first. It's had cave pearls in it for quite a while, probably only since about 2000. I'll show you why in a second. That sample site you'll notice is right next to the road. Those big rocks protect it from some stuff, but they started to turn this into a major haul road about 2008, 2009, and started throwing a ton of debris in here, and my experiments got buried. So fortunately for me, right at that point, the second site developed cave pearls. It did not have them before, and it's far enough from the road not to have ever been affected by the road traffic. So here's what happens in Site A. In March 2006 is the first time I got a good photographer in there and started sampling. Note that nice square piece of carbonate sticking out. By 2009, that square of carbonate, still at the red arrow, was almost completely buried. So in 3 years, the pool aggraded. By the end of the study period in 2014, it had aggraded 20 to 30 centimeters to build a huge platform. This rapid rate of accretion over the time period of the study suggests it probably started around 2000, that these were not something that developed since the 1970s when this area was mined, but that is a more recent shift in the groundwater flow to give the drips. Just at the time when the truck traffic got horrible, I looked at the back wall, and that gave me my second location. In 2007, the back wall was dry and there were no pearls. This drying allowed a flat area to develop. It had not been flat before. It had been on a sloping surface. And that flat area, remember, flat area is required for pearls, allowed pearls to start developing once the drought ended. By 2014, this site was pretty much buried and there were no longer cave pearls forming. The sort of stringy looking things in the 2014 photograph were my experimental attempts that did not really work. I didn't quite have the system down. But you can see the large number of pearls in 2011. That really gave me freedom to sample some areas and leave some areas to sample a little bit later. The rate of precipitation really blew my mind here. I'm used to working in Carlsbad, which is not an active system anymore, so everything's 40,000 years old. This is a picture from 2011 and a zoom in on that little small round, this little rimstone dam pool. It is full of pearls. None of those pearls were there 3 years before. Notice how nice and loose they are. I came back a little over a year later in October of 2012. Notice the not so nice picture, that's mine. And notice how the pearls are completely growing together. I actually sampled those pearls before they glued together because I figured that was my last opportunity to do it. And you can see that the pearls actually start to develop flat sides where they touch each other. Unlike ooids, pearls do not roll over. They are stationary but moving around so that you get growth on all sides. So let's take a look at those layers. On the left is one of the pearls from the back, Site B. The pearl on the right, this one, is the very first pearl I ever cut, and it just about blew my mind when I first looked at it. But let's look at what the layers are. Some are dendrites, nice places where calcite grows very rapidly in sort of tree branching patterns. The white here is porosity. They stay very porous. They are sometimes thicker, and if they are, it's thicker on top, not always. Then there are some very dark brown, almost black, transitioning laterally to very light tan layers. These are so fine you cannot resolve them in a petrographic microscope. And then there are sort of gray laminae, microspar to fine spar. I mostly want to focus in on that brown layer today, but the microspar layers have the same basic pattern. So thinking about how these things form, what would make these different layers? Oh, wait, first, before we do that, let's take a look at the chemistry. We microsampled this with a drill to pull out different layers as best we could. And this is the stable isotope data that we got courtesy of Southern Methodist University. I still have contacts there from my PhD. And note that you really don't see a difference between dendrites or the fine or the microspar. They pretty much all overlap each other. From a mineralogy viewpoint, we x-rayed the same samples, and it is effectively 100% low mag calcite, no variations. I'll show you later, we actually did some microprobe data on some of the variable fabrics, and there's no difference even at the microprobe scale. That says something about what's changing or not changing in the pool. So this little pool, right, I had lots of little pools to pick from. So in 2011, I sampled all of these, and I just took everything in the pool and then made a selection of it. In the 2021 paper, you'll see all the way down to the little tiny pearls that were at the very bottom. These are the larger ones, but they illustrate something I think very useful. All of them have the fabrics. They've got dendrites to one level or another. They've got the brown tan to one or another. They have the microspar. But even pearls that form right next to each other do not have the same details. You cannot compare stratigraphy from one pearl to the next, even though they were in the same pool and developed at the same time within centimeters of each other. That's a very important point, I think. We spent a lot of time before we got all of these together trying to see what variations we could correlate. We can't. So how might these different layers form? Dendrites, we actually have a pretty good handle on. A lot of good work by Jones and others shows that dendrites occur in calcite when you have very high supersaturation rates. So perhaps these represent dry seasons, or perhaps they're the droughts. I think that's some of it. But really, when we look in detail, right, they're too different from one pearl to the next. So remember, this is all from this one little tiny pool. And yet we have different numbers of dendrite layers in different pearls. There is more dendrite in the pearls that are on top. The larger ones are on top. These end up reverse graded in place. So there is some connection perhaps to the air-water interface, which is where degassing is going to occur, and therefore we might get higher rates of supersaturation. But it can't be something that's seasonal or drought because there's just too much variability at the centimeter scale. The fine laminations are a separate issue and quite interesting, I think. So one question is, we've got all these layers, how many? Can you count them? And then over what time period? These pearls are all a few years old, but no longer than that. Even the ones that were collected initially in 2006 can't have been there for very long, given how fast that pool was aggrading. But because we do have a handle on the time, and the very best handle we have on the time was actually the first thing I published from this location. When I first got out there in 2005, I went, ooh, let's start an experiment. And so I took little chips of rock, and it really doesn't matter what you put in this pool, it'll grow calcite. I think if I slowed down, I would grow calcite. So in this particular case, the chip was a piece of the Burlington limestone. So you can see it's a crinoid packstone. I tied it up with fishing line. If you ever wondered what fishing line looks like, it looks like those little yellow things there in that picture. Put it in May of 2006, came back in January of 2007, and realized if I did not sample it that day, I was never going to sample it. So I was like, okay, that's the extent for this experiment. And I pulled it out, and you can see that each of the things is covered, and you can also see the string itself was covered. So 1 to 2 millimeters of growth in a very short period of time. More dendrite on top than on the bottom, but note the layers do go all the way around. And let's take a little closer look at this bottom corner. This is the experimental pearl, that bottom corner. And I want to look first at the laminae. I'll remind you, counted out the number of days this was in the pool was 247 days. When we count the laminae here where it is the darkest, we get over 70. Exactly how many you get depends on what you want to define as, oh, that's a new layer or that's not. And that really makes it very difficult to figure out an origin for it. It's not daily. We know the temperature goes up and down daily. Trucks go in and out daily, but there's too many for this to be daily, right? Or too few for daily, too many for anything else that might be climatic. It's not weekly, it's not monthly, it's not annual, that's for sure. I'll come back to this slide in a couple minutes. The other thing that says it's not climatic is the variation between pearls that are taken from the same exact pool. This was part of why I sampled that pool and got everything in it, so I wanted to be able to compare. And, these two pearls grew during the same time period over the same location within centimeters of each other. Therefore, these laminations must be something very subtle. I did not do the microprobe type chemistry, but I don't think it would matter given the scale on this. This very subtle chemistry within each of the pearls, it seems different when you're a centimeter away. Second thing I want to think about is recrystallization, and remind you, for my definition of recrystallization anyway, new crystals, same composition. We commonly think about it of aragonite to calcite in limestones. Calcite to calcite is also possible. And in this case, it is calcite to calcite. So I use this as my sort of advertising, because what is the question of this pearl is, this is the very first one I looked at. How much of this is primary? And I had some assumptions that it should be mostly primary because of how young it is. It didn't agree with me. And by the end of today, I'd like to convince you that very little of this pearl is primary. I'm not sure how much. So the easiest to see recrystallization is the one that first blew my mind, and that is these very obvious cross-cutting reaction fronts of microspar cutting the fine laminations. They often start in the dendrite layers and go out. They don't have to start in a dendrite layer, but almost all of them do. And you can see very clearly they are cross-cutting the microspar, therefore they must be younger than the laminations. Interestingly, this even showed up in the experimental pearl, right? And this is what I was looking at in 2011, was this incredible reaction front. We polished this thin section, that's where we counted our things, and also did microprobe across it. There's no chemical difference that we could determine at the microprobe level. There's no different isotopically. The contact is an extremely fine line. So this is a reaction front of some sort, going from 1 to 2 micron crystals to microspar. When we started looking in more detail at the curls in the back, where we got this beautiful brown to tan laminations, the question fairly quickly came up, why the color change? And notice that the corners tend to be darker and the flats tend to be lighter, but you can follow individual layers and they clearly change color. In the petrographic microscope, you can't tell. And so we shift to using a scanning electron microscope. And this is where I collaborate most with my colleague, Mike Spilde. He runs the scanning electron microscope lab at University of New Mexico. So I started working on his machine, this older one, and then he got a brand new test scan. And then fortunately for me, Western Illinois got one. And so I continued working on this. This process of looking at thin sections in a microprobe in the SEM brought up an interesting problem that we hadn't realized, and that is the epoxy that is used to glue the sample on embeds itself into all porosity, and it looks identical and also has a carbon signature as biofilm. Since we've been doing geomicrobiology, we wanted to not have that confusing factor. And so we started to use, instead of the thin section, a mirror to the thin section. So take a single cave pearl and cut it in half, take one half off to make a thin section out of, and do a whole image photomonograph. And then on the other side, lightly polish it and then etch it with hydrochloric acid. Doesn't matter how much you etch it, a very light etch will pop what we're after here. To make the two images actually match, since they're a mirror image, I just flip one of them in Photoshop so that I can track reasonably closely what petrographic fabric I'm looking at when I'm looking at the chip. So let's take a look at what we see then. So when we have these dark to tan layers, the tan regions in the SEM 50 micron scale bar are really quite large bladed calcite crystals. 10 to 20 microns wide, sometimes even less than that, but they can be 20 to 100 microns long. You can see here, if you look closely, right, this is all the same crystal, right? And it's actually one crystal going all the way through. And this is what gives these the sweeping extinction in cross polars. But when you etch it, you see these ghost laminations. There's nothing obvious in the mineral that tells us why there's a lamination there. And if you don't etch it and you just look at a polished thin section, you don't see these ghosts. So there's something very subtle in the calcite that reacts to the hydrochloric acid. If you shift from the light layers to the dark layers, here at the same scale, this is seeing both light and dark layers. Let's zoom in and just look at the dark layers right here. You can see that where it gets very dark, the crystals get very small. So that each one of these 1 to 2 micron thick laminae is actually an individual crystal. Again, this is etched. So we're again emphasizing the changes from one layer to another. The darker it gets in the thin section, the finer the crystals in the SEM. You can actually do stratigraphy on these. It's really kind of fun to do. You sort of have to keep track of where you are. So this is a tracking image. I take an overall image in the scanning electron microscope and then keep track on my computer with Photoshop where each image is. So every number here is an SEM image. Just looking at this one layer that varies in how dark it is, you can pretty clearly see that the darker it is, the finer the crystals. And that when you get really light, like right in here, you really don't even see the ghost laminations. You can completely lose in this recrystallization process any evidence of what the original looked like. To really convince you, this is a comparison around one corner of one pearl. So here's the sort of overview image. Yes, there is some cross-cutting stuff coming in here, down here where the white arrow is. What I'm interested in is what happens to the laminae as we go sideways. So over here on the left is an area that in the thin section is very dark brown and has very fine crystals. Go over on that same image, follow the red lines, and you see that it is very coarse bladed spar. And in fact, it all becomes one crystal going the width of the pearl. The black and white arrows also mark layers, in this case, a bladed layer that goes all the way around. Just orient you of exactly how this is changing. And let me remind you, this isn't a pearl that is less than 5 years old. And in fact, when I go to that nice experimental pearl, that one has given many dividends, right? We looked at this originally, and this cross-cutting spar recrystallization jumped out and bit us. What I did not see when I first looked at this was that if you look at the laminations, they are not uniform. They are nice and dark here, and they become lighter and less defined. In the SEM, we get the etched bladed calcite. So here we have recrystallization in a pearl that was 247 days growing. And note that recrystallization is almost in the outermost layer. So this is really almost instantaneous recrystallization in a system where we really can control that time factor. So how much of this is primary? I don't think very much. Maybe the dendrites are. And I say maybe because you cannot tell. The dendrites are very porous. If they recrystallize, it's almost impossible to tell. And if we try to go in with the SEM on a non-impregnated chip, you can't. They just charge. It's almost impossible to look at them for very long. In the SEM, they just glow in the dark. And this is in calcite to calcite, right? All low mag calcite, no changes in the chemistry that we can detect. So what? Why care about recrystallization in cave pearls? Well, they're cool, but besides that, what's a larger sort of issue? Let's think about lime mud. Lime mud, we all know it's recrystallized. It is very fine needles. Again, I'm restricting my definition here to the mud, the clay-sized particles, and that in limestones, it is no longer that small. So we have this mud to micrite translation. We know it's recrystallized. It actually can be very important depending on whether that is a very porous micrite, as in the Cretaceous example from Kazmarek et al., or if it's not porous, and this example of Giesler. But the problem is understanding this process. We see it in the ancient. Tracking it in the modern has proven to be quite difficult. Axel Munich and all very nicely discussed this. I noticed Axel joining. Hi, Axel. The question here, right, is what drives this recrystallization? We know it's there. What drives it? I think they did a very nice summary, which was very convenient to me in thinking about this problem. And they came up with two primary drives. Mentioned a third, couldn't really test it. I'll come back to that third. First is the mineral drive. If we are looking at modern muds, they start as aragonite. Aragonite is much less stable than calcite. A metamorphic person would tell you an aragonite is a high temperature, high pressure mineral, but nobody told, the seafloor that. But it means that if you can dissolve even the tiniest amount of aragonite, that will increase the saturation with calcite, and calcite will precipitate, which will make more aragonite dissolve, which makes more calcite precipitate. And this drive will actually work until you run out of aragonite. How do you make aragonite be undersaturated? How do you make it less stable? The most common is to change the chemistry through microbial decay. This is a nice diagram from the Munich et al. Paper, and no scale bar. And that's because the depth of these reactions varies with the sedimentology. Again, once you start to dissolve aragonite, you will precipitate calcite, maybe right next to where you dissolve your calcite, maybe not. I think in some cases, the aragonite actually goes to the sea. These are pretty good in a sense for explaining modern muds that are aragonite rich. However, it doesn't help me very much in understanding the recrystallization of my cave pearls. There is no mineral drive here. That's why we did the XRD to verify that. This would also be true in the ancient if our muds were calcitic. There is no evidence of microbial decay. As a geomicrobiologist, I spent a lot of time looking, and the Quincy mine stuff has almost no evidence of microbes. And we know there's no changes in the water chemistry. We can't find any anyway using isotopes and chemistry. So there must be another drive. And I think the other drive is what's known, what else is possible, is Ostwald ripening. And Munich et al. Mentioned this, but realize they can't test it very well. Ostwald ripening, though, is very well published on in the metallurgy and crystallography type literature. This is an example of actually Ostwald ripening in calcite, where over 250 days, they went from little tiny, less than 1 micron crystals to quite large crystals. Now, this experiment was done at higher temperature. They actually think they got some Ostwald ripening in a 25°C experiment, but it wasn't as visual. So small crystals have a greater surface area, and therefore they're less stable than big crystals. And so a crystal starts to dissolve, big crystal steals the calcium carbonate, little crystal dissolves more. So it's a solution cannibalization reaction until you end up with crystals that are stable. This is what I think is happening in our stuff here, is that we're starting with 1 to 2 micron crystals, and they are inherently unstable, even if they are calcite, and that what you get out of it is the bladed calcite. If this went over more time, because these are very young, that bladed calcite likely goes to even coarser. But I don't know at what point recrystallization would stop. I don't know where you get a big enough calcite crystal that it's stable. These fabrics are present in pearls that are much older. Now, our problem in trying to understand Ostwald ripening, and this was the recognition by Munich et al., modern sediments are mixed mineralogy, which means that your mineral-driven alteration is there. It is strong. How do you tell that Ostwald ripening is or is not also working? In some cases, you can test this, and there's a couple of examples. I think the most elegant one is this one by Hover et al. In 2001, and what they did was they looked specifically at high magnesium calcite foraminifera, and they looked in the top 30 centimeters of the water column and also did the water chemistry, and they had a coarsening of the high mag crystals within very, very recent foraminifera. So this is an example of Ostwald ripening in a modern sediment. More studies like this, I think, are needed. I like this summary from Axel Munich et al. Currently, there seems to be a gap in our knowledge of what is happening in the top few meters of burial in terms of authigenic carbonate precipitation. And that is the area that studies in deep time successions suggest is critical. if I missed a word, I can't see the end of my slide. I think what we need, and I think what cave pearls help us see, is that Ostwald ripening is probably also going on. And we need more studies where we can control for that mineralogy. Whether it's high mag calcite or perhaps trying to find a modern system that actually has calcite muds, which is fairly difficult. But I think the important factor here is that we need to do the water chemistry and the sediment textures. There are actually quite a few studies that show recrystallization from the water chemistry, but truly geochemists, of which I am not one, typically do not necessarily spend their time on the SEM. I spend my time on the SEM, but I don't typically do the water chemistry. And so we need these kind of collaborations like this study did to really try and pick out how common Ostwald ripening is in the ancient. So let me summarize. Cave pearls grew very rapidly in a mine setting. And it is the mine setting that allows us to do some of the things that we say here, very rapid degassing, and we have control on the dates, right? We know exactly when these pearls started. They are laminated, but laminated with extreme amount of variability, very subtle variations, even within a single small pool. And they are recrystallized. And they are recrystallized in a very visible way in the cross-cutting microspar and in a much more subtle way in the brown to tan. Certainly when I started looking at these in the thin section, I would not have said those very fine laminae were recrystallized. But looking at them in SEM, they clearly are. And that here our only process possible really is Ostwald ripening. And that helps us get this idea that we need to think a little more about this process in other rocks. So sort of our summary here, speleothems often have annual layers, and I'm not questioning those annual layers. They've been very well defined. I think cave pearls are very finely laminated, and other speleothems are sometimes too. Here they are controlled by local variation. I don't want to question annual layers in speleothems, but what I would like to say is don't make the assumption that they are annual or seasonal or climate in any way. One has to design your study to test for alternative hypotheses. And then in thinking about recrystallization, these pearls recrystallize very rapidly. There is no such thing as too young to recrystallize. And the system definitely suggests that looking at lime mud going to micrite, we need to think harder about how Ostwald ripening might occur and how to test whether or not Ostwald ripening is occurring. I'd like to acknowledge my cave photographers. Without them, this presentation would be horrible. You can see my images are nowhere near as good. And I'd like to acknowledge the cast of undergraduate students who did projects with me on this over several years worth of time, and my funding from various Western Illinois University things. One of the nice things of being in an undergrad program is I don't need very much money, and the university can supply that. I'd like to thank Jim Henry for the idea after a discussion at GSA, and Stephen Lokier and IAS for the invitation to present today. Are there questions?
All right. Thank you very much. That was super cool. We already have some questions coming into the chat. So if you have any questions, please feel free to type them into the chat. Tell us who you are and where you're from. Sorry. All right. So our first question is from Maurice Tucker. Fascinating. What about ACC being precipitated first, then recrystallizing to calcite soon after? And what are the tiny holes in the crystals? Negative crystal or microbes?
It may well start as ACC. We don't have the data to show that. What we've got when we pull them out of the pearl, it's already calcite. We haven't got the refinement for that. I was trying a variety of other experiments. Only that first one worked. All I got on my later experiments was dendrites. The holes, remember, these are etched samples. And so what we're etching is something, they are not the right size and morphology for microbes. We do see occasionally a tiny little bit of microbes here, and we see a lot of microbes in our other cave speleothems. So I don't think they're microbes here. They're too small a scale.
All right, very cool.
Get everything.
Um, I have a question since we're waiting for some other ones to come in. Oh, actually, here's another one, then I'll ask my question. This is from Anna. Apologies if I missed it. When the pearls cement together, is it due to or influenced by microbially induced calcite precipitation, as in the use of bacteria that crack um, remediation and soil aggradation? Watching from the UK.
In this particular case, there are no microbes. And we looked very hard for them because cave geomicrobiology is what we usually work on things that we very much have. I do have some cave pearls, but not from this location, that are very much microbial. Here it's just the calcite precipitating because of that very high CO2 degassing rate that eventually they hit each other. And then they get covered in a pool spar, right? So it's just such rapid calcium carbonate precipitation. And I think that really is attributable to the fact that they're degassing that mine. They're rapid speeding the degassing because of the venting of the mine, right? So a diesel truck can go by and you don't smell a thing. That's required by safety regulations, right, as an active mine.
All right, our next question is also from the UK, from Sebastian. They missed the first few slides, so they hope it's not a silly question. What do you think what do you think it is important on stable isotopes? Recrystallization often brings about questions regarding isotope signals. Do we retain the primary isotope signals?
To get a change in your isotopes during recrystallization, you really need to have a change in your water chemistry. These are changing so fast that it's in the same water. And there's no microbial involvement. It's just the CO2 degassing and then Oswald ripening, as I interpret it. And so there is no change in the water chemistry and therefore no change in the isotopes. You would expect it. That's one of the things that we use as do we tell recrystallization. Here there is no change, and I think it's because there's no change in the water chemistry.
For some of the other caves that you looked at, did you see that were like slower for the degassing? Did you see thicker layers or anything different between the mine and the more natural caves?
All pearls are naturally laminated. My feeling, and I'm not sure everybody would agree with me on this, but partially based on what I've seen in this mine, I'm not sure any of the cave pearls from Carlsbad are primary. There's a little bit of primary aragonite in one of the pearls we studied, and you can see it changing, but it's a very, very thin layer. So it's very hard if you don't have any control to tell recrystallization. Those cross-cutting fronts were what I saw the first time. It was like, wow. But this other stuff can be very subtle. I don't see those dark brown to tan laminations very often in my other pearls. But I tend to think if it isn't, mini micrite size, it's probably recrystallized.
Mm-hmm. Very cool. Our next question is from Kate in Texas. Awesome talk. Is there a depth minimum for the cave or the mine? Could the cave just be below the surface?
This mine isn't very far down. It's about 30 meters below the surface. And all you need, and in fact, my Arthur Singleton cave, which has the big, huge microbial pearls, is about 2 meters below the surface. The key for making cave pearls is to have a flat surface. Otherwise, all you get are other speleothems. So as long as the incoming water has sufficient time to become saturated with respect to calcium carbonate, and studies on aquifer says that takes about a meter.
Very cool. Is there, kind of jumping off of that question, is there a depth for the amount of standing water that, is there too much water at some point won't make the cave pearls, or?
There is, and I'm not sure exactly what that is. In most systems, pearls are on the 1 to 2 centimeter size, and deeper than that won't make them. The water drips into water, it doesn't agitate at all.
Mm-hmm.
But there are examples from the literature, Brian Jones's work in the Caymans, and from that Arthur Singleton cave, which I haven't yet published on, where the water depth is more like 10 centimeters.
Hmm.
And you get these big, huge pearls, and they're hugely microbial at the same time. Perhaps because as the water goes stagnant and starts to evaporate, the microbes have the chance to grow. When you look at deep pools, and Carlsbad had, when it was wet, a lot of very deep pools. All of the cave pearls are in water depth less than 2 centimeters. Arthur Singleton and the Cayman says it doesn't have to be.
Hmm.
But I don't know of any that are greater than about 10 centimeters in depth. 10 to 20.
Awesome. All right, our next question is from Jim. Great talk. What about the brown color of the laminae? If not due to organic material, is it likely due to light scattering by fluid inclusions? I wonder if lots of very small fluid inclusions also impacts the surface energy of the calcite crystals, so that there is a drive to recrystallize to calcite with larger crystal sizes, but also fewer inclusions. In the unetched samples, do you see any fluid inclusions with the SEM?
Not that I noticed. I will admit, however, I've not spent very much time on unetched samples, because when I go in them, they're boring. And so I tend to look for other things. The initial crystals in the darkest layers are 1 micron. So I'm not sure what I'd see in a 1 micron crystal. I think the darkness in thin section is the fact that the thin section is 30 microns thick. And if I have 1 micron crystals and a 30 micron thick thin section, I'm getting light going right through 30 crystals. Whereas the bladed, I probably have a single crystal thickness or maybe 2 crystal thicknesses. Sort of the general idea why in thin section micrite is dark. Only here it's almost mini, it is really mini micrite and is almost completely opaque. I'd love to look for fluid inclusions, but I think it's too fine. It's probably there, right? But I don't think I can test that.
Be very interesting. The next question is from John. Any laboratory experiments known that reproduce the same features under controlled environmental conditions?
There is a little bit of cave pearl literature from the 60s that did get some laminations in a lab experiment. It was not properly controlled for biology because we make the assumption in the 60s that microbes aren't in caves because we couldn't culture them. Of course, the first thing that happens when you try to culture a cave microbe is it dies. DNA has shown that caves are full of microbes. I don't know of any more recent ones. I don't really have a lab set up here, so I treated the mine as my personal experimental location. Not quite as controlled. I'd love to see somebody try to make pearls again. I don't know of any efforts since the 1960s.
That would be super cool. All right, so if anybody else
All you
has,
young people.
yeah. If anybody else has any other questions, feel free to put them in the chat. But when the video goes up, I'm sure you'd be willing to take some questions over email if people think of them later on.
Yep. Always interested in seeing. I mostly do speleothems these days, but not exclusively.
Perfect. Well, thank you very much for your talk today. And make sure everybody comes back next month for the seminar at the regular time, 4 p.m. UK time, where it's going to be a student webinar on the volcanic contributions to the stratigraphic record. So be sure to see you then.